So we at the DCIC also created a query tool to query the CMAP data. And this is our version. It's called L1000 CDS square. We used a different method to compute signatures to identify differentially expressed genes. And we have a subset of the signatures. It's from the first 1 million collection of small molecule perturbations, but we also only included in the search only the most significant perturbations. And the search, one of the advantages is that it's very fast, and it's also, we benchmarked it against the Z-score methods and showed that we get more of the correct up and down genes. So we published this tool last year in a Nature journal, and one of the examples to demonstrate that the tool works was an experiment where we received some data from dendritic cell infected with Ebola, and we predicted some small molecules that can potentially inhibit Ebola infection, and we sent a list of the mimickers and the reversers of the signature of the cell line infected with Ebola, and the mimickers seems to work, and this TAP compound, which is a GSK kinase inhibitor, seems to be in a dose-dependent manner inhibit Ebola infections in various human cell lines. And this was a collaboration with people from Fort Detrick, St. Patrick Reed, from the USA MIIRD, which is the research laboratory that they can actually test live Ebola viruses. So this idea of like collaborating and propagating links data and to actually validate some of those hypotheses experimentally is something that I'm really trying to push for. And last summer we had a really great collection of summer students, and one of them was a high school student. And her project was a really simple project. She went to GEO and identified studies where you compare normal and diseased kidneys from diabetic nephropathy. So in diabetes, one of the complications is that you get kidney disease, and there are a lot of studies that profiled kidneys, that part of the kidneys, the glomeruli of a kidney, where you compare normal versus the disease state. And we query all those signatures with this tool L1000 CDS square to find consensus predictions for small molecules that can either reverse or mimic the disease. And our collaborator, Si-Jang He at Mount Sinai, was convinced enough to test some of those compounds in animals, and this is ongoing, but he has some positive results. So this OVE26 mice is a very severe diabetic mice model. And then this is showing with the treatment of this PLK inhibitor, which came out as one of the top compounds that were not known to have an effect. We also predicted some compounds that were already known to be have a positive effect on diabetic nephropathy. And this new compound that is not very well used showed some positive results, and now we're trying to understand the mechanisms of those positive results, and this is actually done in animals. So the ability of the high school student, the Jen Lin, to be able to do this project was enabled by mostly the work of Gregory Gunderson. He was, many of you met him last year. He's now at the computer science PhD program at Princeton. He published two papers, two first author papers, and one of them was this GEO to enrich your Chrome extension that enabled you to go to GEO and click on samples and automatically extract signatures. And then the next part was you can tag those signatures and create those collections of signatures, and then you can compare those collections. And this is an example of looking for glucocorticoid agonists. So all of those mimickers, so all of those columns are studies where dexamethasone was used on to treat various human cells or mouse cells. And then we looked at compounds that can either mimic or reverse dexamethasone. We show that over a third of the molecules are falling into a cluster that have many dexamethasone signatures that were profiled by the L1000. And we also get some novel compounds that are potential mimickers of dexamethasone. And the visualization of this is an interactive heat map. And this is the work of Nicolas Fernandez, and he is also have a poster here today. And he was also enabling this, also the ability to write those, create those interactive reports that give you this analysis of these collections of signatures that are extracted from GEO. We also have a project that uses this query tool for the L1000 to help with reprogramming of MEFs, mouse embryonic fibroblasts, into hematopoietic stem cells. And this is the setting of the program. So Ihor Lemishka's lab at Mount Sinai published a paper where they showed that if you induce several transcription factors in MEFs and put the cells in the right culture condition, after several weeks you can obtain hematopoietic stem cells, which is a big deal to actually get hematopoietic stem cells in a dish and be able to reprogram MEFs to hematopoietic stem cells. And now a new student in his lab is using the L1000 CDS square and L1000 querying approach to identify small molecules that can push the cells further towards becoming hematopoietic stem cells and potentially not requiring the induction of the transcription factors, and also improving efficiency of the reprogramming. So potentially another application for L1000 data is to identify small molecules that can help us reprogram as well as direct differentiation in this regenerative medicine field. So Ali, who joined the lab several weeks ago, was able to first collect gene expression for major cell types, and she has, with the help of Alex Lachman, collected average gene expression for all of those cell lines based on RNA-seq from GEO. And Alex processed all this data, and this is a heat map of the similarity of expression across those cell lines. And then she computed with the L1000 CDS square tool the small molecules that can drive either drive transition from one cell type to another. So now you have a beginning of a map of how you can do those cell transition with small molecules. And also we can do this with transcription factors and potentially start looking globally about how small molecules push cells in various directions. We also have a project with Chris Bassler, who moved from Mount Sinai to Georgia State University, and he's now the chair of immunology there. And he's interested in interferon alpha mimickers. So the HMS links, they actually profiled interferon alpha in various human cell types and submitted it for L1000 analysis several years ago. There are also interferon alpha signatures from GEO that we found 5 studies that were fitting, and then we queried all of this data to prioritize small molecules that could be potential interferon alpha mimickers. And they also got some positive results. There's some 2 NCATs compounds that no one really knew what they're doing, and we got Ajit from NCATs shipped the compounds to Chris, and they got some activation of interferon alpha and interferon beta. And you can see that there's consistency across all of those studies. So this is pretty exciting. L1000 data as well as those GEO data are matching sort of like the same drugs. The same drugs are coming up as the top ranked drugs as interferon mimickers. So Chris decided to do some RNA-seq with one of the compounds that they got excited about, and they also compare, so they use interferon alpha as well as this compound in 2 dosages, 5 micromolar and 10 micromolar in 2 cell lines. So they didn't do replicates, which is pretty bad, but the data look pretty clean. And what we see here is that interferon alpha moved the cells just a little bit on this PCA, and the genes that interferon alpha is inducing are exactly the interferon alpha response genes that are known. However, the compound is pushing the cells. It does turn on some interferon alpha genes, but it's really disruptive to the cells at both dosages. And as you increase the dose, you actually see an even greater change in expression. So the interferon alpha change is very small. So this is sort of like a negative result that should potentially teach us some lessons about getting excited about the fact that a single molecule turn on a gene that we care about. And that's a lot of people are interested in finding what small molecules will turn on a specific gene the most. But this shows you that maybe for this particular compound, it's not the best interferon alpha mimicker. I'm also excited about a very similar project where we collected signatures for ALS, comparing normal muscles and other relevant tissues to ALS and mouse models of ALS, and then prioritizing small molecules that can either mimic or reverse ALS. And there is this company called Psychogenics who has this platform to screen small molecules for ALS using rat model, where they actually image the rats and they use machine learning to gauge various properties of the videos that are collected from the rats treated with the various drugs. So one idea is to actually, and now we are talking to them to test some of those top predicted consistently across studies, small molecules for this. So another direction that we are going with looking at this L1000 data is to visualize all this data. And when you are looking at a signature and you get some highly ranked drugs, it's really critical to know that whether those are unique and what is the space of perturbations and how we can look at the whole thing. So Bruce, you saw this map before. This is the collaboration that we have with HMS to visualize 2,344 signatures for about 100 drugs. And you can see that the drugs cluster by the drug class. So you do see nice clusters of where those 6 breast cancer cell lines get pushed around. And it is by drug class. And the cool thing about this data set is that Peter also measured cell viability. So the blue means that the drugs kill the cells or retard their proliferation, and red means the opposite. Actually, the cells are increasing in proliferation. So just by eye, you can see those triangles, which are those BT20 cells and the HS578 T cells. They actually escape the drug from being from killing the cells, and they actually increase in proliferation. So if you zoom in, for example, on this cluster of this HS578 T cell, those are not T cells, but those are a breast cancer cell type that for this particular group of drugs, the drug actually increase in proliferation, and then you can query the drug, and then you can see that, for example, there are several, these are JAK3 inhibitors, and then we can find the mechanisms and potentially the target using the L1000 CDS square tool. So we were able to scale up this visualization of signatures, and now there is really a transformation in our ability to visualize large scale data sets. And this is what we call a fireworks visualization of 17,000 signatures. And this is coloring by time point. And this is 3,700 drugs, 63 cell lines, 3 time points, 51 doses, and 17,000. And this is the work of Zichen, who is here and has a poster to show this. And this is coloring by cell type. And this is sort of like answering Bruce's question. Can you actually see what are cell type specific responses versus general responses? And I think you guys also showed a plot that shows that some of those small molecules have very cell type agnostic response. And those clusters, like this one, is gathering all of the chemotherapeutics. So those are DNA damaging agents, and many of them are known. But you also get some novel ones in that cluster. So you know you can assign function to those novel ones. These are some HDAC inhibitors that are cell agnostic. And then you have some CDK inhibitors. And these are some other downstream kinases that are all there. And then the upstream tyrosine EGFR type of kinases are clustered in that corner. So, and then these are some mTOR and PI3 kinase inhibitors with some novel ones. So this is not a TSNE or a PCA plot. This is a network clustering algorithm that was borrowed from Cytoscape. But all of this graph is searchable, interactive. You can zoom in and you can explore it and search for a specific drug, or you can submit your own signatures and see where they fall on this plot. So I mentioned how searching for a specific drug that can turn on a specific gene can be very misleading, but actually people want it. Like people ask for an app that give you my gene, and then you can search for a drug that's either going to turn the gene on or turn the gene off. So we have developed also, what started as a summer research project by Kevin Sani, who is an undergraduate student in chemistry at Harvard, and then with the mentoring of Zichen, we created, and the work of Ned He, who has a poster about it here, we created this phone app that it's called Dr. Gene Badger, where you type in your gene, and then it asks you whether you want to upregulate or downregulate the gene. And then we have 3 sources for finding the small molecules: the old CMAP, the Affymetric-based CMAP, the new L1000 CMAP, and then these collections of signatures that we have from GEO, where we extracted signatures for small molecule perturbations across many studies. And then you get a ranking of with various types of sorting of small molecules that can potentially turn on or turn off the gene. So the field in general is moving towards RNA-seq, so Affymetrics. All of the project of collecting signatures from GEO was done by finding Affymetrics assays on GEO and then queried them against the L1000. And as you can see by this plot that was made by Alex, there is a decrease of submissions of Affy to GEO and a huge exponential increase of RNA-seq submissions. So we needed to sort of like move also this extraction of signatures from GEO to RNA-seq, and Alex built this system pipeline that optimizes the resources, uses the cloud. We received a small grant from BD2K to work on the cloud, and he created this system where you have those containers that work as they do parts of this RNA-seq pipeline. Okay. And quickly, Alex extracted all of the human and mouse samples from GEO and is now building a website that you can interact with all of those samples. So this is a visualization of a PCA of 55,000 human samples that you can query, and you can find the samples of interest. This is the visualization of all the human genes clustered using t-SNE based on co-expression across all of those samples. And again, you can find your gene of interest. You can see where it falls next to other genes, and you can potentially find mechanisms for the genes. And there is also a way to get the data directly from GEO through a Chrome extension that was developed by both Dennis and Alex, where you can now install the Chrome extension and get the process data on the GEO page. We're also now able to extract gene, drug, and disease signatures from GEO manually, and this is the work of Troy and Caroline, who manually went and found samples of disease versus normal, drug versus normal, and then were able to give those to Alex, and then Alex ran the pipeline and found the differentially expressed genes for those, and those were added to the enricher software for people to query. You can do a lot of stuff with this RNA-seq data, and one thing that you can do very simply, you can identify average expression across cell lines and tissues, and you can also predict function based on co-expression. So this is, on the left is the average expression across cell lines and tissues, and then on the right, those are predictions for CFTR, go biological processes, and transcription factors that are potentially upstream of the gene. So this is just the method, and Alex can explain you the method of how he predicts go terms. I'm going to skip this and just going to say that we also have a way to benchmark. So Alex uses Callisto, which is a very fast aligner, to align all of those data sets. We wanted to benchmark Callisto versus STAR to see if we get loss in our ability to extract the proper up and down genes. And this pipeline shows you how you can benchmark various aligners, how you can benchmark various differential expression analysis methods, and then you use the data from ENCODE or the CHIA database to try to find matches when you perturb a transcription factor and then measure gene expression after the perturbation, and then the binding sites of the transcription factor upstream of the same gene. So the concordance between those two lists of genes can tell you, or the ranking of those genes can tell you how much your pipeline, how well your pipeline is doing to independently identify the right up and down genes. So we are processing a lot of other data sets besides the RNA-seq data from GEO, and Moshe is creating IPython notebooks for processing all of those data sets. We are starting to build like a library of those IPython notebooks, and those could be potentially on their own publications, and this is the accumulation and contribution of various types of notebooks that we created. And the key thing is now you can start bringing all those data sets together, make predictions using machine learning, and this is maybe Rachel Hodo's poster is talking about those virtual experiments that you now can perform using the data that exists to impute knowledge that does not exist yet, and you can make a lot of predictions about conditions, cell types, and also knowledge that is known about the part of, and machine learning is right in the center of this, and this is sort of like where things are going, where the future is, and basically machine learning is taking over also in our field. And we also have a notebook that you can take the various data sets that have been processed and then build a predictor very easily by just swapping the files that you need. And the last slide is just a cute visualization. So this type of machine learning exercises can be done by a library like TensorFlow, that was provided by Google. Google also with TensorFlow developed this tensor board, which allow you to visualize data using PCA or TSNE or some other methods. And this is the work of Anders in collaboration with MapLinks, where they are trying to cluster images of cell lines. And this is the actual images of the cells are embedded in the PCA plot. And you can explore the images directly and see how they cluster based on the perturbation and their morphology. So this is I would like to thank all the members of the lab for all their work and for coming here. Nick has just got a very, Nick Fernandez got a big job, and he's going to be leaving us in July. But we have this guy over here that we're trying to recruit. His name is Felix Fernandez. So still negotiations are going if he's going to be accepting the offer. And so that's it.